Hello students, today we will see about theory of linkage. Theory of linkage is given by T.H. Morgan. We know that Morgan is considered as father of experimental genetics. Now, Morgan has done his experiments on Drosophila, which we commonly call as fruit fly, and proved his experiments on the linkage in the year 1910. In 1910. So his first experiment was done in 1910 year. Now, what are the characteristic features of linked genes? Linked genes are present on same chromosome in a vertical pattern. For suppose if this is a homologous pair of chromosomes, then the genes will be present either away from each other on the chromosome, on the same chromosome, or they can also be present very nearby to each other. So there are two possibilities of existence. So basing on that also, the tendency to maintain their parental combinations will be dependent. So linked genes occur on same chromosome in a linear pattern, and linked genes have the tendency to maintain parental combination. When will be there a chance of maintaining parental combination? When the linked genes are very nearby to each other, then only there will be more chance to maintain parental combination. But if the linked genes are away or far away from each other, then there is a chance of crossing over and there is less chance to maintain parental combinations. It means that when crossing over happens, there are more chances for recombinations and variations in the characteristic features. Now, strength of linkage. As we have said that when the genes are away from each other or linked genes are far away from each other, then there is more chance for crossing over. So basing on that concept, we can say strength of linkage is inversely proportional to distance between the two genes. It means that as the distance is increased, as the distance is increased, then linkage will be decreased. So what are the factors that are affecting crossing over? If distance between linked genes is more I am representing as arrow mark towards upper side, then chance of crossing over will be more. And if temperature is increased, then also chance of crossing over is more. Next, as the genes or chromosomes are exposed to X-rays for more time, then also the chance of crossing over is more. In case of any type of living organism, as the age increases, then the chance of crossing over between the genes and or chromosomes will be less. It means that as age increases, the capacity of variations will be less. So as the age increases, the chance or the capacity of variations will be less. Very important point to know is, in case of male Drosophila, crossing over is absent. This point is important to learn for you students while answering the statement model of questions. So the fifth point we can mention here after the fourth one. In male Drosophila, the crossing over between genes is absent. Okay, so next, let's see the types of linkages. There are two types of linkages. First one is complete linkage, second one is incomplete linkage. Now, the linkage in which genes always show parental combinations, that type of linkage is called as always the parental combinations only should be expressed. That is called as complete linkage. Second one, incomplete linkage means if crossing over has occurred between the linked genes, then the linked genes of two different or two separate chromosomes, then the type of linkage is called as incomplete linkage. So we'll write some of the points. The linkage in which genes always show parental combinations only. Next, such type of linkage will not exhibit the variations or crossing over or new combinations of genes. This type of linkage never shows new combinations of genes. It means that crossing over is completely absent. Next, here, if crossing over is not able to happen, it means that the genes are very nearby to each other. Genes are very closely arranged on chromosomes. Basing on all these points, we can say that complete linkage is, we can say it is very rare situation in the evolution or nature. So, such complete linkage is considered as very rare in nature. Very rare in nature. So, what examples can we take in means basing on this complete linkage? We can take male Drosophila. As I have said before, the crossing over is completely absent in case of Drosophila, and also female silk moth. It also does not show any crossing over, and it is considered as a complete linkage genes containing organism. In case of incomplete linkage, here there are more chances of new recombinations. More chances of new recombinations. Here we can take examples like female Drosophila. Next, we can also take maize plant, and we can take human being or man. So these are the two differences between complete linkage and incomplete linkage. Linkage group. All genes on a single chromosome, they form one linkage group. Now, in case of heterologous set of chromosomes, in heterologous set of chromosomes, the linkage group is present on one chromosome. Now, here, what is meant by heterologous set of chromosomes? In case of heterologous set of chromosomes, the alleles are on different loci. Alleles are on different loci. Such type of chromosomes are considered as heterologous. We know that homologous type of chromosomes also, those are having the related identical gametes to pair, but here it is not that. It is considered to be opposite to that of homologous chromosomes. We will see some of the examples also. So, some model of statement-wise questions are seen recently. In that, the number of linkage groups basing on the type of ploidy of the organism, if asked, we should consider number of linkage groups. The number of linkage groups of a species are always equal to haploid set of chromosomes or haploid number of chromosomes. Example, we can take one example. For suppose, Drosophila is having 8 number of chromosomes. Eight number of chromosomes. If a question was asked, what is the linkage group in case of Drosophila? As I have given here statement that linkage group is always equal to haploid number of chromosomes, we can say linkage group number is 4. Linkage group is 4. Take some examples to represent the total number of chromosomes which we consider as 2n condition and haploid number of chromosomes and what is meant by linkage group number in different types of organisms. If we take one example, that is human beings. We know that the 2n condition is 46. If 46 is considered as 2n condition, then haploid number of chromosomes are 23. Next, I told you that linkage group and haploid number of chromosomes are same. It means linkage group also will be 23. Next, chromosomal pair also 23 pairs of chromosomes only. Second example, if you take Drosophila, total number 2n condition is 8, then everything will be half of that only. Third example, Pisum sativum. Here the total number of chromosomes are 14, then haploid condition and linkage groups are half of that. Fourth example is corn plant. Here 2n condition or the total number of chromosomes are 20, then half the number will be equal to haploid number of chromosomes or the number of linkage groups. According to NCERT, page number 84, we have seen the total diagrammatic representation of Morgan's work on Drosophila considering the body color, that is brown color or white color, yellow color, and also the eye color as red and white. So Morgan has done two types of crossings. So Morgan, has done two types of crossings. In cross 1, he selected two characters in Drosophila, that is body color and also eye color. In cross 1, he selected two characters. Those are, one is body color, second one eye color. In case of body color, the dominant genes are represented with capital Y. We know that contrasting alleles or genes will be existed for any type of character. So in case of body color, the dominant one or brown color of the body is represented by capital Y and or capital Y plus I have written here, and normal recessive character, that is yellow color of the body is represented without plus mark. Okay, so Y plus, it indicates the natural type of body color in case of Drosophila, that is naturally existing in the nature. In case of eye color also, we are representing like this. This is indicating red color of the eye that is naturally existed in the nature, and this is represented as white eye. So Morgan has taken the parental combinations, but I said you in the nature only brown colored bodied Drosophila and red eyed Drosophila are normally existing. Then how in page number of 84 NCERT we have seen the parental combinations as white-eyed means male and female Drosophila. How can we see such types of combinations? So first, before that diagrammatic representation, what Morgan has done, that we will see. Morgan has taken normal, natural type of allelic forms in the Drosophila only. So here, parental combinations taken initially by Mendel are, we know that linked genes are present on one chromosome. So this part is indicating the type of two different types of alleles but on the same chromosome. This line we are indicating as chromosome. Now on one chromosome, we know that always homologous or one set of chromosomes will be present. So when dominant type of alleles are present, immediately on their paired chromosome, recessive type of alleles are also present, but recessive type are not expressed in presence of the dominant type of alleles. He crossed. So this type of chromosomal arrangement indicates the Drosophila is having brown body. So this one indicates brown color, this one indicates yellow color of the body. In the same way, W plus, this indicates red eye color, and normal without plus mark, this indicates white-eyed or white color of the eye. So here when a Drosophila is having such type of allele arrangement, it means that that Drosophila is having brown color body and red color eye. This is crossed with male. So we will consider this one as female Drosophila. It is crossed with Y plus, W plus chromosomes. We know that in case of male organisms, in case of Drosophila, we represent hemizygous condition, means one X chromosome and one Y chromosome. So here this is considered as Y chromosome. This is considered as X chromosome. So this is X and this is Y type of chromosomes. Morgan has done cross here like this, and then he obtained some different types. Before starting his experiment and to obtain white-eyed male Drosophila, Morgan also sprinkled many types of mutants that are responsible for causing mutations in any type. He did not expect only the type of mutation in male or female separately. Just he had sprinkled some types of diluted bases, acids, some types of chemicals to extract the mutations and observe the mutations in case of Drosophila. He just did an experiment initially just to see whether any type of mutations will happen or not. But his experiment was success that he has seen first time white-eyed male Drosophila. And we are practically diagrammatically representing the same type of work done by Morgan here in form of this type of arrangement. So the first work done by T.H. Morgan includes the parental combinations like this. We extract the gametes, means parental gametes, from this type of arrangement of chromosomes. We will get the combinations as, this is one gamete with two different characters, another gamete of male, this is of female, this is of female X, this is of male X, and also another gamete with no types of any allele forms in that or genes in that. So in F1 generation, he has seen purely when the cross is between X of female and X of male, he has got the offsprings like this. We call them as homozygous type because both are of dominant featured alleles only. We call them as homozygous. Homozygous, red-eyed, brown body. This organism is female. Next, another organism or another offspring. When this is crossed with the Y chromosome, X of female and Y chromosome of male is crossed, then what are the combinations? This is X chromosome with Y. This is indicating brown body with a red eye. But since there is Y chromosome here, we can say this is male organism, male offspring we can see. Now, so what term can we give here? Here I have included homozygous type of allele arrangement. Then what term can we give here? We can give here it as hemizygous. Hemizygous. Now, when the X of this one is again crossed with this, then what type of offspring we will get? On one X chromosome, we get Y and W. On other X chromosome of male, we are seeing Y plus W plus. It means these both are dominant alleles. Since there are existence of one dominant paired alleles, one recessive paired alleles, we can say this is heterozygous type of organism or allele arrangement. But since both are X chromosomes, we can say heterozygous, brown body, white-eyed female. Now, another combination we'll see. X chromosome of a female crossed with Y chromosome. That is purely like this. And Y. Now we can say there are no existence of the dominant type of allele forms here. So this indicates again hemizygous only. Okay. So since this is X and this is Y, we can say this is hemizygous condition. But it is indicating white eye with yellow body. White eye with yellow body. Now, from these offsprings, we know always how we are doing the experiment. We are taking the F1 offsprings and self-crossing among them only. Now, Mendel has done his experiment according to page number 84 in your NCERT textbook. If you see, what type of F1 generation are selected by Mendel? Mendel has selected purely white-eyed and yellow body female. White-eyed, yellow body female. But here we are not seeing that type of arrangement. So this is given in your NCERT. But when we have done cross, we are not seeing such type of this one offsprings or the type of allele arrangements. Then what should we do? Now we will self-cross some of the offsprings among themselves and we will see whether we will get those pure homozygous type of white-eyed and yellow body female or not. So let's take this type of combinations and we will do self-cross. We have got here male Drosophila with white eye and this one yellow body, and here we have got heterozygous type of female. Let's cross between these two and we will check whether we will get the type of white-eyed yellow body female or not according to your NCERT textbook. Crossing. Few offsprings from F1 generation. So let's take heterozygous brown body white-eyed female and cross with white-eye yellow body hemizygous male. This is male. So let's cross that. Y plus W plus recessive Y W. This is considered as female. This is crossed with. Again I am telling you these both are nothing but offsprings obtained in the F1 generation. This is crossed with hemizygous male, that is white-eyed yellow body Drosophila. Now, if we have to cross this, we will get a F2 generation and let's extract gametes from this type of combinations. What type of gametes we are getting? We are getting one gamete like this, another gamete like this, another gamete like this, and Y chromosome. So this is X of a female, this is X of a female, this is X chromosome of male, and this gamete is of male Y chromosome. Coming to F2 generation, the X of a female, now if crossed with the X of male chromosome, male Drosophila, the combination will be Y plus, means dominant type of alleles of female and recessive alleles of male. This is considered as heterozygous female because we are seeing the contrasting alleles here. Heterozygous female Drosophila. Another combination, X of female with Y chromosome. We get the allele combinations with dominant one and Y chromosome. This is considered as hemizygous male. Hemizygous male, but here it is of red eye only. Next, another combination. X chromosome of a female, but here purely it is recessive characters or recessive alleles only. So when it is crossed with X chromosome of male, we get the combinations like this. Means on both the chromosomes, the alleles are of recessive type. It is considered as homozygous female. Now, when this same recessive alleled X chromosome is crossed with Y chromosome, then we get white-eyed male because there is Y chromosome. So again, this is hemizygous, but this is male. Hemizygous, because hemizygous is the condition in which there will be one X chromosome only of the pair of XX chromosomes. So this is the combination. Now, what is given in your NCERT textbook? They have taken this one as parental combinations and they have crossed with normal hemizygous red-eyed male. The cross is taken between these two types of offsprings. It means that in case of page number 84 NCERT textbook, the original parents from the nature are not taken as it is. They are made some changes in their allele arrangement and crossing over might have happened. And so we got white-eyed yellow body female and red-eyed, this one is naturally existed. So in your NCERT textbook, they have taken the parental combinations from F2 generation. So they have taken the parental combinations as purely yellow body, white-eyed females which are crossed with red-eyed and brown body male. This is indicating dominant one which are present, naturally existed one are this type of combinations. Let's take out gametes from this. We can call them as parental gametes. So one gamete will be having purely recessive, another gamete also purely recessive. These are of X chromosome of the female cross with X chromosome of male, again with a dominant species, and Y chromosome. This is X and this is Y chromosome. Now let's cross here. In F1 generation, we cross, if these types of gametes, we get this type of allele arrangement is representing heterozygous female because there are one dominant allele arrangement, recessive allele arrangement also in the both X and X chromosome. So this is indicating as female organism. This is heterozygous female offspring. Now, another cross we'll see. This is X of female and Y chromosome. This is considered as hemizygous, but the alleles are of recessive type, male organism or male Drosophila. Now, another combination. Again, we are getting X chromosome of male and X chromosome of female. Again is this heterozygous female. Drosophila. Now the last combination is purely white-eyed male. This is X chromosome of female with Y chromosome. Again this is hemizygous male. We can say this one is of brown body and red-eyed female. This one is yellow body. Next, white-eyed male. This one is brown body, red-eyed female. This one is yellow body, white-eyed male. So these are the combinations that we got in F1 generation. Now, let's take some of these organisms of the offsprings of F1 and do the self-crossing so that we will check what are the parental combinations. So, what are the types of offsprings and what is the percentage of parental combinations. So here Mendel has again taken only some of the offsprings from this, and here I need to tell you the same type of allele arrangement in the XX chromosome of a female can also be written as like this. Next, the Y alleles of Y chromosome of males, male Drosophila, can be written as simply like this. Now, when Mendel has crossed this type of heterozygous female from F1 generation with a purely white-eyed female, white-eyed male Drosophila, this type of cross is also called as test cross. We got only two types of offsprings from the F1 generation. So the heterozygous type arrangement is like this. So when such type of cross is done, we call it as test cross. So what is meant by test cross? Any of the unknown type of genotype is crossed with purely recessive type of alleles. Such type of cross is called as test cross. Means test cross is to know the unknown genotype of the parental combinations. So what are the gametes from F1 generation when we are doing like this? Y, dominant one, purely this is one gamete, and here recessive one is another gamete. Now, actually these have to give only these two types of gametes. But what happened in case of Drosophila, when the parental combinations are giving rise to gametes, they also gave two another types of combinations of alleles in the gametes like this. One dominant and one recessive. This is heterozygous type of combination we can call, but that is quite different, the type of normal heterozygous type. So if we see how these have been obtained, this one and this one, we can also write this type of allele combination as Y plus Y and W plus W. Means dominant and recessive one we can express like this. If we extract the gametes like from this one, it will be obviously one combination or two dominant alleles, Y plus W plus, one type of allele combination. Next, another type of allele combination, that is Y plus W, which I have written here. Another type of allele combination, recessive and one dominant, recessive allele and dominant allele of another character, which I have written here. Next, another type of combination is both recessive alleles are pairing. So these are the type of alleles that I have written here. Now, we already know that in case of dihybrid, from one offspring we get four types of gametes, allele combinations in the gametes. Now, when these are crossed with recessive male, this is X chromosome and this is Y chromosome. This is one gamete, this is one gamete. When these 4 types are crossed with same type of X and Y type of gametes. Then what type of offsprings we are getting? If we cross this one, Y plus W plus, this is X chromosome of a female that is fused with X chromosome of male. Then the combination is like this. It is considered as heterologous female. This is one type. Now, when the same type of dominant alleles are paired with this one, Y chromosome, then we get X chromosome of a female and Y chromosome. This is considered as male Drosophila. That is natural one without any recombination. We can say this is a parental type. Now, another one. Here, okay, this is fused with the both. Next, we'll take the second one. Y, which is a dominant allele and one recessive allele, X chromosome of the female, now crossed with X chromosome of male. Now, this is considered as recombinant because the gamete itself is representing recombination because both are not dominant alleles, both are not recessive alleles according to the parental combination. If you take the parents, if you see, both are recessive here. Here both are dominant on one chromosome, both are recessive on one chromosome. Here, but here, if you see, one is dominant allele, one is recessive allele. So we can call this one as recombinant. Now another combination. Y plus W means X chromosome of female that is crossed with male. So this is another, again we can call this one as recombinant. Since it is XX chromosome, we can call recombinant female. This is Y chromosome, we can call recombinant male. Now again we will see this bottom 2 types of allele combination in the gametes with the recessive male Drosophila. I am taking now this one. Y chromosome with the recessive alleles on the X chromosome of a female with pure recessive alleles. Now it is crossed with X chromosome of the male. Again we are getting same type, means like that of parents only, means recessive female. There is no recombination, means both are of recessive alleles only. Another combination. This is crossed with Y chromosome. X chromosome of a female is crossed with Y chromosome. Again this is of parental combination. Now the last one. X chromosome of the female which is having one recessive and one dominant allele now is crossed with X chromosome of the male. Now this one we call as recombinant female. Again this is crossed with Y chromosome. We are getting again recombinant male because of existence of Y chromosome. Now I am writing here the terms like this is parental type. This is also parental type. This is also parental type. This is also parental type. And here 1, 2, 3, 4. Actually here we should not consider exactly 4 number of parental type 4 number. Drosophila, the number taken by Morgan are more in number. There is no count. There might be some 100 or 50 or something, suppose. But whatever the number that are means approximately taken by Morgan, we can consider that one as 100%. So out of the total number of Drosophila that are present, Morgan has observed more percentage of a parental combinations only within the 2 characters like body color and eye color. So here, when Morgan has considered the percentages, he has observed more number of parental combinations. That is parental combinations. He has observed 98.7%. And the recombinance percentage, he has observed recombinance very, very less in a number, that is 1.3% only. If we recall the Mendel's dihybrid cross, we can see there 16 number of offsprings are present. out of that 16 also, we will see the ratio, phenotypic ratio in case of Mendel as 9:3:3:1. Here, these middle 3 and 3 are indicating the recombinants. 9 and 1 are phenotypically representing the parental type of characters or traits. If we consider here also, we have to take out of the total 16 number of offsprings that are taken in the dihybrid cross according to Mendel, more number are parental combinations comparing to the recombinants. So, here in case of Morgan's experiment, comparing to this 10 number also, still some more number are giving parental combinations. That some more number is equal to 98.7% are being expressed as parental combination type of alleles, and very, very negligible or less number are considered as, means 1.3% are nothing but recombinants, are considered as recombinants. So by this experiment, you should understand that whatever the diagrammatic representation given in NCERT textbook is not from the original natural combinations, but those are already previously crossed, and from the F2 generation, the offsprings are taken and that are considered as the parents. And then test cross was done among them by taking the recessive type of males, which are giving white-eyed, this one, characters. And in the F2 generation, means from the F1, one type of offspring, heterologous type of offspring was taken female. It is crossed with this type of recessive male. Okay. So, please don't consider here they are given only 8 and 8. But no, we should not consider the only whatever the number given there. We should consider the percentages. Okay. Parental combinations and recombinant combinations percentages. And then you should remember that the cross from F1 generation is done. That type of cross is test cross, not the regular type of self-crossing. So, by this experiment, by taking two types of characters, Mendel has hypothesized or thought that eye color trait is connected with X factor or sex factor or X chromosome of the male. Next, this thought in turn also led to an idea of genetic linkage. Means when more number of parental combinations are observed, it means that there is more linkage within the genes or alleles which are present on the same chromosome, means linkage. The distance between the allele forms is very less, so that the parental combinations percentage is more. If the distance is more, then crossing over chances are more, so that we'll get recombinants more.